Hey A&Pers, this is Dr. Jones checking in. This is A&P 1 Lab 3 Video 2. So we are still on the muscle physiology models. We're on page 25 in the note set. And we are starting with the big myofibril model. So the myofibril model starts with the second box. So we're going to start with the sarcolemma. Remember there is, well, if you got to this part in lab, I will have told you that there is not a rough connective tissue covering on this model. We start with the smooth sarcolemma, the cell membrane. So sarcolemma, and then you can see here the mitochondria. And let me zoom in on one. There we go. Hey, I can zoom. So you can see that there's an outer membrane and inner membrane with little folds called cristae, and then matrix inside the inner membrane. And you can tell by what they've done with the cut here that they clearly mean for this to be a mitochondrion. Okay, I'm going to zoom back out. Okay, there we go. So this going the other direction, just under the sarcolemma, is going to be the nucleus on the model. All of the rest of these are mitochondria. We've got another cut one up here to show that all of these directions are mitochondria. And then we need to talk about the myofibril, which is this thing. So now we have zoomed in again. So we started with the myofibril this size, zoomed in to a myofibril this size, and now we have zoomed in again. So one myofibril, and here's a half a myofibril, and then we'll have a part of a myofibril here. Okay, attached to the sarcolemma, you can see this indentation leads to this, was painted green, tube down the middle. Tube down the middle is the TTubule. And touching the TTubule on either side are terminal cisternae. The TTubule with the two terminal cisternae that are touching it, those three together, are a triad. Now the terminal cisternae is a part of the sarcoplasmic reticulum. It's part of the sarcoplasmic reticulum, but it's specialized, so it has its own name, just like your hand is part of your upper limb, but it's a specialized part, so it has its own name. Still part of your upper limb though. So terminal cisternae are part of the whole sarcoplasmic reticulum, which I usually call a lacy sweater sleeve, and I refer to the terminal cisternae as the cuffs. So, myofibril, T-tubule, sarcoplasmic reticulum. Okay, so now we're moving to this part up here. No, let's do it this way. Okay, we're going to adjust. We're not going to adjust. Hey, fancy. Okay, let me lock that little adjuster so I don't knock it off. Okay, so, oh look, it's a zigzag. What do we know about zigzags? They are Z-lines. What are Z-lines made of? Actinin, A-C-T-I-N-I-N. And here's another zigzag. So that's also a Z-line. And Z-line to Z-line is a sarcomere. So between my thumbnails is one sarcomere. So attached to a Z-line are thin filaments. Thin filaments are made of actin, A-C-T-I-N. They don't act out, they act in. Okay, so here's the length of the thin filament. In the middle of the sarcomere, M for middle, is the M line. And this M line has been dissected so we can see its crystalline structure, which we don't entirely care about, but there it is. Attached to the M line are the thick filaments. The thick filaments are made of myosin. So the thick filaments continue out to here. So between my thumbs, everywhere there are thick filaments is an A band. Everywhere there are not thick filaments is an I band. In the center of the sarcomere, on either side of the M line, is an area with only thick filaments and no thin filaments, and that's the H band or H zone. And then here, where the thick and thin filaments overlap, is a zone of overlap. And here's our other zone of overlap, because there's thick and thin filaments overlapping. So the length of the thick filaments, A band. What are the parts of an A band? Thick and thin filaments both, zone of overlap. Thick filaments only, H band. Line down the middle, M line. More overlapping thick and thin filaments, zone of overlap. So there are 2 zones of overlap in each A band. No thick filaments, only thin filaments, I band. Z line to Z line, sarcomere. Okay, let's do that on another model. I have to move the camera again, just so. Okay. Zooming in. All right. The dark splotches between my thumbnails are A bands. The line down the middle of an A band is an M line. The light splotches between my thumbnails are I bands. Line down the middle of an I band is a Z line. Z line to Z line is a sarcomere. Sarcomere, 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 sarcomere. On our other model, zoom out. Hey, there we go. So the dark splotches are A bands. Line down the middle of an A band, M line. The light splotches are I bands. Line down the middle of an I band, Z line. Z line to Z line is a sarcomere, sarcomere, sarcomere. So on these 2 models, you can see A bands, I bands, M lines, and Z lines. You cannot see the H bands or zones of overlap. Okay, so I will come back and talk about the pretty model. So this has been A&P 1 Lab 3 Video 2. Education must go on. Dr. J out.